The overall goal of the following experiment is to construct gene-targeting vectors using subcloning plus insertion. This is achieved by transforming the BAC containing E. coli with a recombinering plasmid. The second step involves generating asymmetric phosphorothioated insertion cassettes and subcloning plasmids by polymerase chain reaction. Following the addition of arabinose, the GBA-A recombinering proteins are expressed, and the terminal modified insertion cassette and subcloning plasmid are electroporated. The desired DNA sequence is simultaneously captured from the BAC and modified with the insertion cassette into the subcloning plasmid.
The main advantage of this technique over existing methods like standard recombinering is that gene-targeting vector construction can be performed in a single reaction.
To begin, design oligos for the insertion cassette and the subcloning plasmid. Design each oligo such that it contains 180 base pair homology arms flanking the genomic target site, and 20 base pairs of specific priming sequence for the insertion cassette or subcloning plasmid. Next, determine the orientation of the genomic DNA insert of the BAC clone using a web-based tool like CloneDB. Establish the direction of replication from ORI-S by checking the map of the BAC plasmid backbone used in the BAC library construction. Add two terminal phosphorothioate bonds to the 5' end of the oligo that is opposite the direction of replication on the BAC clone. Next, add a 5' phosphate modification to the reverse oligo. First, stab a sterile pipette tip into the BAC agar culture and inoculate 5.0 ml of lysogeny broth. Grow the culture at 37 degrees Celsius for 5 hours with shaking at 200 rpm. Next, chill 10% glycerol solution, microcentrifuge tubes, and electroporation cuvettes on ice. Cool a refrigerated large centrifuge and microcentrifuge to 4 degrees Celsius. After incubation, determine the optical density of the culture using a spectrophotometer, measuring absorbance at 600 nanometers. Prepare the electrocompetent cells when an OD600 reading of 0.3 to 0.8 is reached. When the culture is ready, spin down the cells in a 50 ml centrifuge tube. Wash the cells with 1 ml of chilled 10% glycerol and spin again. Perform these wash steps a total of 3 times. Resuspend the cells in a total volume of 50 microliters and add 10 to 200 nanograms of the recombinering plasmid. Obtain a single cell suspension by pipetting up and down several times, and then transfer the cells to a pre-chilled electroporation cuvette. Next, place the cuvette into an electroporation device and electroporate the cells. Immediately recover the cells in 1 ml of LB and transfer the cells to a 50 ml centrifuge tube. Grow the BAC culture at 30 degrees Celsius for 2 hours with shaking at 200 rpm. After this time, add 9 ml of LB to the recovered culture. Grow overnight at 30 degrees Celsius with shaking at 200 rpm. To incorporate the homology arms into the insertion cassette and the subcloning plasmid, perform PCR using the long-modified oligos prepared earlier. Alternatively, linearize the plasmid template using a restriction enzyme. Choose one that cuts outside the PCR amplification region and is heat inactivated. Set up the PCR using a high-fidelity hot start DNA polymerase system. Prepare a PCR master mix and perform thermal cycling according to best practices. After the reaction, analyze PCR products by agarose gel electrophoresis. Load 1 to 5 microliters of each PCR onto a 1% agarose gel. Next, purify the PCR products using a PCR purification kit. Quantify the PCR amplified DNA against a known set of DNA standards or by using a nanodrop spectrophotometer. After an overnight incubation, dilute the BAC GBA-A culture 50-fold and prepare a separate culture for a negative control. Grow the diluted cultures at 30 degrees Celsius with shaking at 200 RPM for 1 hour and 50 minutes. Next, chill all recombineering materials and equipment to 4 degrees Celsius as demonstrated earlier. Prepare a 10% weight per volume solution of L-arabinose and filter sterilize through a 0.2 micron syringe filter. When the OD of the BAC culture reaches the desired level, proceed with inducing the recombinating proteins. Add 200 microliters of the 10% arabinose solution to 10 milliliters of the BAC culture to achieve a final arabinose concentration of 0.2%. Include an uninduced culture to be used as a negative control. Transfer the cultures to a 37 degrees Celsius shaking incubator and induce red expression for 45 minutes with shaking at 230 RPM. Spin down the cells and wash with 10% glycerol 3 times as shown earlier. Add 600 to 1000 nanograms each of the insertion cassette and the subcloning plasmid to the recombinating reaction. Include a vector only and vector plus single insert control to check the recombination proficiency and integrity of the vector. Next, obtain a single cell suspension and electroporate the cells as shown earlier. Allow recovery in LB media at 37 degrees Celsius for 1 hour for multi-copy plasmids, or in 10 milliliters of LB at 37 degrees Celsius for 3 hours for BAC vectors. Plate different dilutions of the recovered culture on dual selection agar plates and grow at 37 degrees Celsius for 16 hours. When ready, pick colonies into 5 milliliters of LB containing the selective antibiotic and grow overnight at 37 degrees Celsius. Next, use a column purification kit to prepare mini-prep DNA. Introduce the appropriate restriction enzyme and then identify the clones containing the expected fragment sizes by gel electrophoresis. Finally, perform DNA sequencing across the homology arms and insertion cassette to check for oligosynthesis errors. In this example, restriction digestion analysis of P2RX1 EYFP insert containing clones showed the expected pattern, indicating that they contained the correctly assembled knock-in vector. This schematic illustrates the concept of back trimming using subcloning plus insertion. Analysis of PCR products confirmed that cassettes were correctly incorporated. In addition, when positive back clones were analyzed after adding the appropriate restriction enzyme, the expected pattern was obtained. Restriction digestion analysis of 12 ZRSR2 SPI clones showed correct vector assembly in the majority of the recombinants.
After watching this video, you should have a good understanding of how to construct gene targeting vectors rapidly using subcloning plus insertion.